has to be a research paper. It has to have methods, results, right? Conclusion.
Doc, do you really care specific journals that these papers come from?
No. As long as it's a research paper. There are a lot of good journals out there. I don't want to bias anyone in particular. It all begins with the search keyword, and from that, I have a list of some really nice titles that define, especially if you want to go by and then look at the abstract.
And your course one would be primary papers.
Well, primary meaning.
That is the original study to that, not a review of that study.
Right. Yes, it's not a review. It has to be
Or not a secondary study caused by that.
No, that's fine too. I mean, there's study to study, right? So because usually if it's a secondary study, it's not a repeat. It's usually a discovery or an addition. So that's fine too. Okay. Yeah. It doesn't have to be the original. No, you don't want to see the paper that discovered the CD04 process, right? And that's not the one we're looking for. Because there's many that have been done beyond that, right? So the key is you want to pick one, and we want to introduce the topic that's specific about what that particular research paper is. And that's the learning process. Because I want you guys to understand that, as I said, research papers are wonderful textbooks, and that introduction part is really valuable part of introduction. And then the second part that's a learning process is think about how discoveries happen, right? And so every paper has a reason for that. So that's the point of once you make the selection, that's one of the questions I'll always ask when I'm in grant. Your class presented a paper. Why did they do that? Okay? And so if you read that paper, you'll realize, wow, I understand why they did this. Beyond the big picture, a little more specificity begins to make sense. So you develop that part of yourself as well. Because as you go out, become professionals, doctors, researchers, you really have to begin to understand how that process works. So it's really, really wonderful. So it sort of takes you to the next level, right? Right now, most people are at the level for finding information, Wikipedia, right? That's kind of the extent. You want to go beyond that, okay, a little bit further than that. Say, okay, I need to start looking at research papers and have a good collection, and then you start to develop that process. And if you go down the road of research, whether it's clinical or basic research, that fundamental process in which they write those papers is basically how you develop your career in the future. So it's a really good time to begin to learn this if you haven't really done this before. So, and then just beyond that too, just simply understanding science, right? Look at the news, think about it. You're at the level now where you can say, well, the news says this, but then if you start to do a little research beyond Wikipedia, right? You pull up a few really interesting journal articles and read them and say, well, actually the media wasn't quite right on that. Interesting. So you can get the truth because the research papers allow you to do that. And it takes you beyond, as I said, beyond Wikipedia. That seems to be it's a really easy source, right? Search, boom, Wikipedia is probably the first thing that comes up. And there's nothing wrong with that. I think it's a good place to start, particularly if you're really unfamiliar with the topic. Go ahead and read that. Kind of introduces you to what this is, right? It helps. But then the details, you really have to go beyond that because you'll get yourself in trouble and just rely on that. And certainly, of course, if you write a formal paper, never reference Wikipedia. Absolutely not. Not a good thing. Okay. That's not a good thing. All right. So I hope you brought notes from Monday because we're going to finish that. And then we'll transition to today's notes. There's a little piece there that we didn't really have a chance to talk about, so we're going to talk about that today. And then sum it up, and we'll move on to the next topic. What's that? I didn't.
The title one is
Uptake systems.
Uptake systems on Monday topic, right? Uptake systems. And then today is high energy molecules. Sorry, I'm hard to hear. It's what happens when you get old. Terrible. All right. Uptake systems. Unlike you sometimes, with technology, it's work quickly. All right, so here we go. So the screen will come on in a minute. But I can go ahead with it and just kind of restate some things regarding uptake systems. Okay, so recap the uptake system lecture. Okay, we introduced 3 major systems, right? And so we talked about why this is important. So we're talking about how bacteria as our model, why they're so successful at growing and causing disease as one example, right? Or why they're so successful at surviving in soil, providing rich nutrients which plants can then grow and then farmers are happy, right? So there's a lot of reasons why we want to understand why bacteria grow the way they do. And one of the ways that we can understand this and improve on this to make things easier for our lives here on Earth would be to understand some of the details that drive this ability of cells to divide and grow successfully, right? And one of the ways is to think about how they internalize nutrients, food, okay? And so there's some interesting ways that we discovered that involve the use of energy and the volume of energy. We've broken them down into 3 categories. Okay, so we talked about those, but I also emphasize that remember not just energy, plus or minus, like the protein, presence of the protein, right? Meaning the membrane now has a protein. Facilitated fusion has that. Active diffusion systems have a membrane, and the protein and the membrane that are associated with this. Okay. Also, I emphasize, besides just plus or minus energy, was modification, right? So that's what we're going to talk about today. So when a nutrient starts from the outside of the cell and it internalizes itself, sometimes that molecule doesn't change. So it's not modified, just simply stays the same. We can use glucose as an example. Glucose is on the outside, it enters the cell, it's now still glucose, okay, unmodified. But then sometimes glucose, as it internalizes, can change. Okay, you consider it a subtle change, but in terms of chemistry, it's actually a huge change. And so if glucose enters and now you attach a phosphate to it, okay, and so now glucose is now called glucose 6-phosphate. That's a modification. So when you attach something to a nutrient, you've modified it, okay? And that's a pretty significant thing that happens in cells. And it's an active system, but it's a modified process. And there's a specific discovery system that we call the phosphotransferase system, better known as the PTS system, okay? So it requires, again, lots of energy to do this. So let's look at what that is, okay, in terms of the system. So I gave you a nice diagram here talking about the system. So we call this, your textbook calls it group translocation. Okay, that's just another way of saying essentially what I just said, that molecules undergo a modification process. So how does that happen? Well, in systems like bacteria, this system actually has a certain level of specificity. It's really quite interesting. So when you think about it, there's different types of sugars that are food nutrients, right? There's glucose, there's mannitol, there's sucrose, the list goes on. So some of those sugars have very specific receptors, protein receptors that they can bind to. Okay, so that's the level of specificity that can exist. So because of that, it's really interesting to think about what are the processes that drive this mechanism. So we discovered years ago that when cells have this specificity, in other words, when do they decide to use glucose and not lactose, for example? Is right? And this is going to introduce the very last part of this point that we'll get into more in class in terms of regulation, but it's a good idea. It's a really good place to make the connection. Okay, so if you haven't been introduced to this concept of how do bacteria choose between using glucose or lactose, because they're both excellent sources of nutrients, right? They're not going to use them equally. They're going to use one before the other. Okay, and if you remember, we call this catabolite repression, or it defines that dioxide growth curve. And it'll make sense later, but this is a good place to think about that. So this process is associated with that. It's the ability to choose one sugar versus the other. Okay, and it begins with specificity. Okay, how does that happen? Well, with this energy producing system, one has to ask the question, where's the energy coming from? Okay, well, the energy is this molecule called PEP. And that will transition us into the next lecture. But just keep in mind, the energy molecule is PEP. So that's where the energy is coming from. And it supplies this by delivering a phosphate. And that phosphate is transferred from that molecule to E1, which is a protein. And that protein attaches the phosphate to a very specific amino acid. In many cases, it's histidine. A very specific amino acid in that protein is what binds the phosphate. And so then that phosphate is moved from that protein to another protein called the HPR protein. Again, histidine usually is the choice amino acid. So there's always a really specific amino acid in these proteins that are going to bind this phosphate. So it's just delivered from one protein to another until eventually it interacts with some proteins that are closely associated with the membrane. Okay? And in this case, if it's destined to bring in a glucose molecule, glucose specifically binds to that receptor, not that one, but this one. Okay? And what happens is it's waiting for that protein to change its conformation. And how does that happen? Well, when proteins change conformation, that usually requires energy. And it usually requires modification of the protein. So how does that modification process? Well, if you attach a phosphate to it, that's one way you can change these formations. Okay, so that's driving this change, and ultimately it allows a channel to open, so the glucose now has a nice little hallway, if you will, right? Channel to then enter again into the cell, and doing so, that phosphate molecule has its energy system that then allows a bond to form in which now the phosphate attaches itself to the glucose. And then it enters, yes?
Okay, yeah, so that's what I was confused about. Figuring it's either it goes to this complex where glucose can go through, or a complex where mannitol.
Yeah, and the specificity is the key. It starts with specificity. Okay. So, in other words, the mannitol will never bind to this, and therefore it will never enter with this association.
So does the glucose encounter this this complex first, and then the phosphate comes, or is it sort of just because it's chaotic in the cell, it kind of happens together, like you were saying earlier? Not random, but in a way.
It's constant. It's what we call concentration dependent. So in other words, these are always there. And whenever you have high glucose, you have a lot of these. And when you have low glucose, you may have less of this, right? So that's the way cells operate. Again, nothing is all or nothing, right? It doesn't all bind or doesn't bind, and systems are not always on completely or completely off. There's always a level in which this happens, right? And so when the level is low, we say that's off. Or in this case, when the level is low, we say they don't enter, right? So it's real, just think about how nature works. It's all about just concentrations, okay? So yeah, so what happens is then the system, and the part that's different too, remember, this part here is conserved. All these proteins conserved. So all cells have these. These proteins here are specific for the sugars. Okay. So if you can imagine, you got two separate proteins. One looks like this and one looks like this. This is your glucose receptor one, and this is your mannitol one. Okay. So that's how different they are. So that's where the entry and the eventual change that will occur as it enters the cell. So the modification key piece here is now that it accepts the phosphate, now changing it into a glucose 6-phosphate. And remember that phosphate came from that high energy molecule at the very beginning of the process. Okay. And so now the link here, and I'm not going to test you on this, but I'm going to go ahead and just introduce you to this because it's really important to think about this. So what does this have to do with cells choosing between glucose and I'm using lactose because the Lac Operon is the key regulation that you guys should be familiar with, right? The Lac Operon system simply means it chooses glucose first, it uses it up, and then it switches to lactose. So that's the Lac Operon system. So what happens here is that when glucose is available in large amounts, this happens, and then nothing's different. But then when glucose is low, what happens is this protein that has the phosphate, rather than delivering it to glucose, it can't do that efficiently because there are not a lot of glucose there, right? So what happens to this protein that has the phosphate? It actually activates another enzyme in here called adenyl cyclase. And what that does is it produces a activator called cyclic AMP, if you remember that. And cyclic AMP binds to another protein called CAP. And that complex binds to a promoter, which then turns on gene expression. Okay? And so that new gene expression would be producing the genes that now metabolize lactose. Before that, it didn't use lactose. It used this system. Okay? When this drops, it activates another system, which then turns on gene expression for the second sugar. And that's kind of how the system works. So it's really interesting how at the processes here, you can then regulate gene expression based on that connection. Okay? So just remember that. I'm not going to test you on it this exam, but it'll come up later. It's a real good place to be thinking about connecting some of these systems. Because you should have been taught that Lac Operon system, right? You should have been taught diaxial growth curves. Right? Remember the growth curve goes, you probably remember it. Here's the growth curve. It grows, it uses glucose. Glucose is used up, stops growing. Those systems turn off, Lac Operon turns on, and then starts growing again. Right? That you should have been taught. Okay? So that ability to switch sugars has everything to do with this system here in terms of whether it's used completely or whether that phosphate is then used to activate another system that turns on genes that metabolize lactose. Okay, so that's the connection. So those active, modified uptake systems, this is the question, are important for supporting regulatory systems such as glucose-lactose utilization. Just that statement I would like you to understand. Okay, the details of regulation will come later. Say it one more time. The importance of this GPS system, which is an active, modified system, is critical for defining how bacteria decide to use different sugars. Another way to look at it, right? So that's the key right there. It's not the genetic regulatory process, it's the mechanistic process. Okay. So that I do want you to understand. Okay. So does that make sense to you guys? Okay. Any questions? Yes?
So could it possibly, for example, like glucose and lactose, could it possibly use both at the same time? Is that how you said like?
Yeah, it's always like that. Remember, it's a ratio, right? So you're always going to, when it starts to use lactose, it doesn't mean it never uses glucose.
Yeah.
It just means that amount of glucose entry is now so low that when you measure this, it appears lactose is the only thing there, right? And also another way you think about it is when glucose level is low, it doesn't necessarily mean they're completely gone. There might always be just trace amounts there, right? Which means the system is so operating at such a low level that it may be non-detectable. So we define it as it's off, it's not being used. It's not really true state, it just means it's so low that it's non-significant. Whereas the more significant one is the lactose utilization, because you have a lot more proteins, everything's active, and it's using that system, and that's what you can measure.
However, those proteins, the transport proteins, those are never going to come out of the membrane.
Yeah, they're always going to stay in there. So we'll talk about this too in the semester. It's called a protein secretion system or a translocation system. So it's how do proteins get into the membrane? We'll talk about that. But yeah, once they're there, they're permanent. Yeah. And they're destined to go there. It's really interesting how that happens. We call them type 1, type 2, type 3 systems, etc. all the way down. We know 6 of them now. I'd say in a few years we'll probably have 7, and we'll just keep discovering some of new systems. So it's, but yeah, we'll talk about how they get there. That's an interesting thing to think about. Okay, any other questions about this last active system? Right, there's 2 kinds. There's unmodified, which we started out, and a good example of an unmodified would be the siderophores. Okay. You think, well, hey, how's that differ? The chemical siderophore binds to an iron, and it brings it in, so now you must have modified the iron. Well, it's not quite the same thing, because basically the only thing a siderophore does, and a lot of times this is what happens in bacteria, so that's why we don't put it in the category of modifications. Siderophore binds the iron. In bacteria, what typically happens is, yes, it then binds the receptor, it begins to internalize itself, and then when it gets to that other side of the membrane, the siderophore releases the iron. Okay, and what happens? The iron changes its oxidation state too. It goes from what we call a ferric, just for your information, an F+3, and then it binds the siderophore, enters, and becomes an F2 ferrous, which is the difference between the two is one's not very soluble, and one's soluble. And so when it enters, it's a nice soluble form, and it's released. So the iron itself is not really modified. In other words, there's nothing attaching to the iron. Remember, the definition of modification is you're attaching something to the molecule. Glucose, you're attaching a phosphate. Iron, you're not, right? It's just entering, the oxidation changes. So that's what I would define as an unmodified system. But it's still active, right? So it still binds the system. ATP has to hydrolyze. Internal it goes, not modified iron. With glucose, binds to the surface, requires an energy source. In this case, it's not ATP, it's PEP. Enters, it's modified because it now has an attached phosphate. There you go, that's the difference right there. You need to understand that. Okay.
So I use glucose in the situation of both modified and then also unmodified. So does that mean it's going to be uptaking glucose in both ways?
Yeah, you can actually have unmodified glucose, but typically in bacteria, this is the primary way in which they enter. So when they get into the cytoplasm, it's this start of metabolism is really interesting. It starts from glucose 6. We learn in the textbooks the glycolysis pathway starts with glucose, right? Well, in bacteria, usually it starts with that second step, glucose 6. And of course, you can go backwards. That glucose 6 can then convert to the glucose if it needs to, right? But that's kind of interesting. So it saves a step. It's conservation. Your book talks about another reason why it does this is that if you want to keep the amount of glucose in your system and not have it diffuse outward, that by changing its form, it now is basically locked into the cell. So in other words, well, if it enters this way, what keeps it from going back out that way, right? Well, this is specific for glucose. It's not specific for glucose 6. So now it's glucose 6, so it's not going to bind to this, find its way to go out. So it's locked itself in there by changing. So that's another way to think about why do they do this. Okay, keeps it in the cell. Secondly, it's efficient. You save a step in metabolism. Okay, so there's some really interesting, and that's what we call conservation, right? Cells always find really interesting ways to save energy to be the most efficient way to do things, right? There you go. So you now are developing some understanding of, if I ask the question, how do cells develop this efficiency? What are ways? Give me some examples of how that happens. There you go, right there. Glucose production, right? Put that in your notes somewhere. Don't forget it. Those are all good things to remember, things to hold on to. All right, so let's go on to the second lecture, high energy molecules. Okay, so this is a really interesting topic that I really think is important for us to understand, because it has everything to do with thinking about, the book talks about, as we think about microbial metabolism, okay, which is basically metabolism and anabolism, right? You know those terms. And thinking about beyond the ability of nutrients such as glucose to be defined as a source of energy. Because glucose is a source of energy, but glucose is many things too. It's a source of carbon, right? And it's a source of electrons. But if you think about some of the detail events that happen in metabolism, it's really important for you to understand and ask the question, what are some specific molecules in the cell that are critical for supplying energy that drives microbial metabolism? The answer is not going to be glucose. Glucose has many roles. The specific molecules, you should think about it and say, oh, which ones are they? This is what this lecture is about, right? So we're going to talk about ATP, we're going to talk about CTP, UTP, PEP16. Okay, those are all examples. Those are key pieces that you really should be familiar with in terms of what these are. So because they're associated with supplying energy, we call them high energy molecules. You'll see this in the literature all the time. They'll mention, so-and-so, such cells had at a certain level of high energy molecules. So if you see that, you'll immediately understand they're talking about one of these molecules that was introduced in class, right? So the level at which these is key. So let's begin with the very first one. Think about how that would be important. First of all, just a couple of key terms. I just added this morning. So go ahead and write this down. The key I want you to think about is this lecture has to do with energy conservation. How does it conserve the energy? And how does it use the energy? And so these high energy molecules really help you to define that. Somebody says, well, how does a cell save this energy? How does it conserve it? High energy molecules. How does it use this system? High energy molecules, right? So that's what we're going to talk about today. So that's the key. All right, so how does that happen? Well, these are the ones that are on the list that are, I think, the most critical in understanding at least the basic processes of metabolism. Okay? You probably are familiar with them. Obviously, ATP. Okay? The other nucleotides, maybe not so much, but this is a good time to learn their association. And then the last three have seen, and probably just need a reminder of exactly where they are. So the key that I'm going to be having you guys learn is that think about not only just the molecules, but think about what they're associated with, the pathways. Okay? So if you can envision in terms of how you learn metabolism, you learn metabolism based on, well, I learned glycolysis, right? So I kind of envision this pathway of glucose going through these processes, and then at the end it produces pyruvate, right? That at least you should remember. And then during that process, I sort of remember ATP having to be used, and then ATP maybe being produced, right? Remember that part of it somewhere. And then also I kind of vaguely remember another molecule called NAD being used as well. So that's sort of the level, the point at which you should be. But I want you to be a little bit more specific about that, and we'll get into it in a second. Yes?
Are we going over the electron change export?
We are, just a little bit, just to introduce that. Okay. So the connection would be, what's the connection of these NAD systems when we call them high energy? Connection is the electron transfer chain. Okay. So we just start from the very top, ATP, and that's really the primary molecule that's found in a lot of different chemical reactions. Okay, they supply the energy to drive a chemical reaction. And so because of that, and all the others fall in the same definition, is that we like to use the word coupling, which is another word for connecting. There's two processes in chemistry. There's what we call the endothermic and the exothermic, right? Exothermic is the release of energy, endothermic is the taking in, right? So that process has to be connected to create a cycle. And a molecule that has energy and can release energy, conserve energy, has that ability to connect it. So it's a coupling molecule, connects the two systems. Okay? And so because of that, what we begin to understand is, okay, if this is a really important coupling molecule that allows the connection of the two processes and it drives metabolism, then the question will be, what part of metabolism do these molecules participate in? Okay, that's all I want you to begin to understand. Okay, so the first one would be, if I were to ask the question, ATP, give me your best example of where this might happen. Again, it happens in a lot of places. The answer is not going to just simply be, it happens in metabolism. That would be a zero answer. Okay. The answer would be glycolysis. Okay. Then I would say, okay, give me a specific place in which that might occur. Well, you know glycolysis, right? You've seen it. It looks like this. I'm not going to ask you to memorize it, but certainly you should be familiar with it. So you ask the question, okay? The answer to the question is ATP, high energy molecule, is associated with glycolysis. Okay. And specifically, it's associated with the generation of ATP, or the use of ADP to drive the reaction involving glucose 6 as one example. I'm not going to ask you all of it, but I do want you to know, providing one very specific enzymatic step in glycolysis. If you want to pick the first one, then that's probably a good one to remember, or you can pick the second one, where you're talking about fructose 6 converting to fructose 1,6. Okay. Either one doesn't really matter to me, but I really want you to understand the specific chemical reaction, because remember, this is the energy that's driving the enzymes to then convert that molecule to its product. Okay? And it requires a high energy molecule to do that. All right. So that's the extent I want you to think about this. Okay? Also, just to get a little bit more specificity in there, I do want you to also think about the same question and say, okay, these high energy molecules, what other pathways are important in terms of what they support? The guanosine triphosphates, it's not metabolism, it's not the glycolysis. One of the best examples would be protein synthesis. We call this translation, right? So I want you to be familiar with that. And of course, images are powerful. That's translation, right? You have message RNA, you have to have ribosomes, and once that's there, you then have a transfer RNA that brings in the amino acids, right? And builds the protein. Okay? So the specific part is this. The energy molecule GTP, when hydrolyzed, that energy now enables the ribosomes to assemble themselves. Remember, a ribosome is basically a large RNA protein complex. Okay? And they're pieced together early on to form this one large piece. Okay? So when those complexes come together, that requires energy to create those bonds to form. Okay? The energy that's required for this system is the GTP. Okay? It's not ATP, it's GTP. And that's how specific it is. So it's really interesting to think about it. What drives translation? Lots of things. It could be the amount of amino acids. It could be having a message. And it also could be level of GTP, right? If you don't have GTP, you're not going to have assembly of the ribosomes. If you don't have ribosomes there, translation is not going to happen, right? That's the question I'll ask. So let's think about this. If the cell doesn't grow well, and evidence shows that it has something to do with protein translation, and it has nothing to do with the level of transfer of amino acids, give me another reason, another possibility. You know the answer, right? You could say, well, it might have to do with the level of high energy molecules, specifically GTP, because I know it's important for assembling ribosomes. That's why this bacteria is not growing so well. That's the point I want to make. So write that down on your notes, because I love to ask these questions about, let's think about why bacteria is growing so well or not growing so well. Okay? And the answer is not going to be always simple. Saying, well, there was no food available. Absolutely, you'll get a 0, might even get a -10 for that. Not a good answer, right? We're talking about some really good information here that you gotta use. Okay? Yes?
Is there a reason why this process uses GTP over ATP?
Specificity. Exactly. It has to do with, again, that protein and the amino acid that it prefers to bind with is more, the affinity is much higher for GTP than it is ATP. Exactly. Everything's about affinity, right? Binding and having the best fit. And nature has created a variety of proteins that allow them to bind to different kinds of molecules, right? And that variability is what makes them so powerful. But also, as scientists, it makes it really interesting because we haven't even come close to figuring all of this out. But the key to figuring this out is going to be the key to curing a lot of diseases or making things efficient so we're not paying so much for fuel. Or what do we do with all this waste that's causing toxicity, right? It'll come down to our ability to understand some of these really interesting details. So we're just scratching the surface. It's really quite fascinating. Okay, so that's the GTP. And then the last two, CTP. It's a lipid is basically what makes the membrane, right? Cell membranes. So if it's the gram-negative or gram-positive, you've got that cytoplasmic membrane. Think about how those lipids are made to make that membrane. Well, that process involves heavy use of this CTP as a source of energy. That drives the building of a lipid that ultimately becomes the membrane. Pretty cool stuff. It's not ATP necessarily, but CTP plays a really important role. And the third one is, you should make the connection now. And we've talked about peptidoglycan synthesis. Okay? Peptidoglycan synthesis actually starts from glycolysis, fructose 6. That's the key intermediate. That ultimately builds itself to become a NAG, and that becomes one of the sugars. All right, so it goes through all these steps. So that fructose 6, you're eventually forming this NAG sugar. I'm calling it the pre-NAG. So the pre-NAG would be, you can call that fructose 6. First, one of the intermediates in glycolysis. So as it goes down that pathway, UTP hydrolyzes itself, it becomes UDP. That's the energy that supplies the conversion of this pre-NAG to become the full NAG that we know. Okay? So you've got a UDP, right? That's the carrier. Now it's the carrier. It's carrying that to the NAG, which now goes from the lipid 1 stage to lipid 2, right? Then it's flipped across and so on and so forth, right? So there's a really important step in biosynthesis that UTP plays in terms of the source of energy. Okay, once it loses that phosphate, it becomes a UDP. What's really interesting, it's now what we call an activated carrier. Okay, and so that's what, if you think about it, that's how it's all assembled. Okay, so those are the connections I want you to make. ATP with glycolysis specific, chemical reaction shown. CTP lipid synthesis, membrane formation, UTP peptidoglycan synthesis. Okay? And then the last few, this one here, PEP. Let's think about it. Where's that? PEP is right here. Called phosphophenylpyruvate. As that converts itself to pyruvate, that's the amount of energy that these molecules have to make ATP. So let me just say a little bit about that. So in order to produce a high energy molecule, or a high energy molecule is special because it has a fairly high level of energy that allows it to then give the energy to something, right? But then think about it, right? If something is so highly touted as being a high energy molecule, then what makes that high energy molecule? Well, if everything's associated with energy, then you have to have something that has a little more energy than that, right? And that's really what it comes down to, is that PEP has more energy available, therefore it has the ability to produce this primary high energy molecule called ATP. That's the only way it works. You can't have a lower energy molecule making ATP. It has to be a high energy molecule, and there are such molecules that do exist. You know, PEP is one of them. Okay. The other one is up there, 1,3-bisphosphoglycerate. That's another very high energy molecule. Because of that, it has the ability to make ATP. Okay, so that's one way to understand how the system works. Is that yes, ATP is primary. It's used all over the place. Doesn't necessarily mean it's the highest. It just means it's nature has chosen to use that. So what's interesting is that in order to make it, you have to have a higher level of energy molecules to supply that. And within metabolism, there are some key places in here that we call substrate level phosphorylation that allows that process to happen. So that's what I want you to be familiar with. The question is, what other high energy molecule is required to make the high energy molecule ATP? Those two, PEP and 1,6. So you'll find that PEP not only drives the production of ATP here, we already talked about it, right? We mentioned it in group translocation, right? What brings glucose in? Beginning of the lecture. That's the high energy molecule right there. So it actually has some really interesting associations with mechanisms. So keep that in mind. And then the last high energy molecule to think about would be these nucleotide systems that we call NAD and NADPH. Okay, so we'll start off with NADH. Okay, these are associated with a reaction called redox reaction, reduction oxidation reactions. Okay, so what does that mean? Well, it just simply means that the ability of these molecules to deliver an electron from itself to another molecule. Okay, so it's a donor and there's an acceptor process. Okay, that ability to transfer an electron is associated with what we call redox potential value. And so every molecule has a redox potential value, its ability to deliver an electron. These molecules have an incredible ability to do this efficiently. That's why nature has chosen them to do this. Okay? And so when they do that, then that ability to transfer an electron from itself to an acceptor, the difference in the redox potential value determines the amount of energy that it can produce. Okay? So in this case, it's not directly making chemical energy like ATP. It's making what we call proton motive force energy. Okay? So how does that happen? Well, what happens is this is the electron transfer. So if you're familiar with that, it makes sense, right? NAD delivers that electron right here to these enzymes that then accept the electron. In doing so, that hydrogen that's attached to it, the NADH, which is the hydrogen, is now transferred across the membrane, creating this ionic charge. In this case, we call it a proton motive force. If there's sodium, it would be called a sodium motive force, right? But it's proton, so it's hydrogen. So it's that gradient. And that gradient is potential energy. It's not chemical energy, it's gradient energy, another form of energy. And that's what this particular high energy molecule is associated with. And so when that happens, it then produces that potential energy in which those hydrogens then make their way back into the cytoplasm through this enzyme called an ATPase. And that enzyme drives the reaction in which ATP diphosphate becomes a triphosphate. And that's the energy that now makes ATP. So ATP can be made in a couple ways here, right? From a very high energy molecule, like PEP, right, in glycolysis, or it can be made from a redox high energy molecule based on this ion gradient formation. So I want you to think about that. So the association with high energy molecules, we've just covered some of the key places in microbial metabolism in which this happens. So that's a real good foundation to have. As you go forward and begin to think about how cells operate, these are the key molecules that are really important, that play really important roles. And you'll see it more and more in the literature. The more you read about this, it'll come up. And say, yeah, I remember we had a nice lecture on that, right? So that's why they're so important. And they come up over and over and over again. And there's so many complexities that's really fascinating, and you can make a career out of it. That's the key here. So make sure you understand that connection. And I think images are the best way to go. So you're saying, well, how should I study? Well, again, ATP, glycolysis, CTP, membrane formation, lipids, GTP, translation, PEP, ATP synthesis and glycolysis, or translocation, right? In ADP, electron transport chain, making ATP. And that's the connection right there that you have to be able to be very, very familiar with. Okay? All right, how are we doing? Okay. Okay, we've got group presentations on Friday. Okay, I'll send the papers to you today. We'll get yours today, I guess. And then read that, be ready for questions on Friday. And then I think that's it, guys. Yes?
You didn't do a test flight?
I'm working on it. So it's looking good, by the way. I'm pretty happy with what I see so far. Seriously.